The overall goal of this biochemical protocol is to generate nucleosomes with differentially isotope-labeled sister histones for use in mapping post-translational modification reactions simultaneously on both sister histones.
The overall goal of this biochemical protocol is to generate nucleosomes with differentially isotope-labeled sister histones for use in mapping post-translational modifications simultaneously on both sister histones. This method, in combination with high-resolution techniques to map post-translational modifications such as NMR spectroscopy, addresses key questions in chromatin biology, like what mechanisms contribute to the formation of asymmetrically modified nucleosomes. The main advantage of this technique is that it can be easily established in a biochemical laboratory because it combines standard nucleosome reconstitution protocols with common protein purification methodologies.
Two pools of histone H3 will be used for octamer reconstitution. Nitrogen 15 labeled H3 that contains a polyhistidine tag at the amino terminus, and carbon 13 labeled H3 that contains a streptavidin affinity tag at the amino terminus. Begin this procedure by dissolving the lyophilized histone aliquots, including the two types of histone H3, in unfolding buffer. To each tube that contains about 5 mg of histones, add 1 ml of unfolding buffer and mix by pipetting, but do not vortex. Keep the tubes on ice for 30 minutes to allow complete unfolding. Next, measure the absorbance at 280 nanometers to determine the exact concentration of each histone. Mix the core histones to equal